Yes, Chandu, we are on live now on YouTube.
Apurva, may we begin the webinar?
Sure, please.
Okay. Alvin, can you please start the webinar? I will start it. Alvin, can you hear me?
Yes, shall I start it?
Yes, please. Yeah.
Yes, we are on live now.
Yes. Okay. So people are joining. We will wait around 2 to 3 minutes before we begin. Hello, everyone. We will begin in about 2 minutes. We are waiting for people to join. So we are nearing about 90 people now. So may I begin with the instructions?
Yeah, please go ahead.
Yes. Okay. Welcome everyone to our Science Setu webinars. Since plenty of you must have attended the previous webinars, the instructions will be short this time. Please know that chat will not be enabled for any of the attendees. So if you have any comment, any issue, if you cannot hear anything, etc., and if you have, most importantly, questions, kindly put them into the Q&A box. You can just click on the Q&A box and put the question in there, and we will address it as soon as possible. Also, for those of you watching on YouTube, please type your questions in the comments. We will be monitoring the YouTube comments, and we will relay those questions back into the Zoom room. With that, I hope you enjoy this webinar, and I will hand it over to Professor Apurva Sareen, Director of inStem. Apurva, over to you.
Thanks so much, Chantakant. Good morning, everybody, and a welcome back to this series. I also understand that we have students joining from medical schools in Chennai, so a very warm welcome to that audience as well, and we look forward to your continued participation. As we have noted throughout the series, that while this gives us an opportunity to engage in times when one-on-one meetings or larger meetings are not possible, I think we are all eager and looking forward to a time when we can go back to something closer to life as we knew it. But just, I think it behooves us to just stop possibly and think about what we can do as individuals with an interest in science, which is the shared interest that has brought us to this particular forum, to think about what can we do as individuals. The pandemic is obviously overwhelming in its scale, but I think it just remember that a very simple precaution like wearing a mask, especially a double mask if you are outside, is one of the best barriers that we know to possibility of exposure and contracting infection. This is also extremely important for those of us who are performing the roles of caregivers to those who might be unwell or affected in our homes and in our community. And with our knowledge and our awareness, we are urging everyone that we meet and everyone that we encounter in different fora that please do take this precaution. And as scientists, budding scientists, people with a passion in science, interest in science, it's really our responsibility to do this very small little bit towards stopping the spread of infection. The other, of course, is isolate and test if you feel that you've been exposed. There is absolutely no stigma with this infection. It can be handled, it can be managed. The most important thing is to give yourself the knowledge that if you have it, you must get tested and you must then take whatever steps are required. So with just that little reminder, which we are going to reiterate across all the full series, because we feel it's a very important small step, but of great significance if each of us did our bit, we'll now turn to the actual the seminar for today. And it's my very great pleasure to welcome my colleague, Praveen Vemula, who's an investigator at inStem, for the seminar today. For those of you who've been with us throughout the series, you'll notice that we've made a small change in the sequence of talks, and we've done this to as a sort of a celebration of National Technology Day, which was on 11th May. We felt that in fact, we have with us a scientist who really exemplifies the possibilities that exist in technologies and the ability to find resolution using technologies and knowledge in physiology to find solutions for unmet needs. And so it's with great pleasure that I at least am looking forward to Praveen's talk, and I hope that you too will find a lot to learn from what he tells us about their work. Praveen's work really straddles the fields of material science as well as physiology, and therefore it's a very, very powerful combination of approaches. that has manifested and I think has been the driving force for much of the work that he has executed. So with that, over to you, Praveen, and thank you very much for being here. I'm going to stop sharing and hand over to you.
Thank you, Apurva. Yeah. Good morning, everyone. So, as you know, recently we have celebrated our National Technology Day on 11th of May. So we thought on this occasion we can discuss about the roadmap for developing the technologies which can potentially have huge societal impact. So if you take a overall developing technologies to bring it to the end user, this entire process called translational science, and there are variety of various definitions are known for the translational science, but a widely accepted and very standard definition is developing the technologies which can address the unmet clinical needs and reach to the people to improve the quality of life. So that's the overall motto and aim of the translational science. And if you see this entire translational science, it has a very distinct phases. And typically it starts with the preclinical development, which is also known as a T0 phase. Perhaps this is the most important stage in the clinical translational science. So this includes, identifying the right set of the problems and coming up with the ideas and do a lot of basic research, come up with innovations and develop the solutions and test these ideas and with the series of in vitro experiments and so on, and show the proof of concept and validate this efficacy of the technologies using appropriate disease models in animals. So that is the overall series of steps involved in the T0 phase. And the next phase is a clinical development phase, which consists of a T1, T2, T3, T4 phases. And this is primarily involve all the clinical trials, phase 1 to phase 4. But more importantly, if we see, it's not necessarily that all the technologies that have been developed at T0 phase, all of them, they don't enter into the clinical development phase. Rather, a very seldomly, a very small portion of the technologies that developed in T0 phase actually enters into the clinical development phase. If you look at this closely, there is a huge gap because it's not a natural transition that all the technology that are developed at T0 phase, they can directly enter into T1. But rather, there is a huge valley that exists between these two phases that is known as a classical valley of death. And it's irony that the majority of the scientific breakthroughs or innovations of the technology developed at T0 phase, they remain at the laboratory. They're not able to cross this valley of death to reach to the clinical development phase and subsequently reach to the people, right? However, to build this gap, one can actually efficiently establish the entrepreneurship program that enables the translation of these technologies into de-risk them and make them ready for entering into clinical development phase. So this is the overall roadmap and the path for doing entire clinical translational research. So what we do is today, we will focus much on T0 phase because this is one of the most important phase that decides how successful your entire translational program going to be. And because if you see in T0 phase, there are multiple, especially 3 key factors majorly influence developing these impactful technologies. Now, one is identifying the unmet need. It sounds very simple, but it's not as simple as it sounds because there are numerous unmet clinical needs exist. There are problems to solve. Then the question is which one to pick because everyone has finite of so resources, time, and so on. So you need to wisely invest all these efforts to pick the right problem to solve, right? So for example, if you are a scientist who are developing drugs, if your field is drug discovery, so which disease you pick to develop the drug, right? For example, you put all your resources and develop a drug for a headache, right? Because as you know, like it takes like hundreds of million dollars of investment and 5 to 10 years of development time to develop any drugs. And of course, like any drug discovery, then you can generate a lot of interesting biology and a lot of knowledge you can generate. But the success of the technology will be measured with a different yardstick in the translational science, right? For example, like if today, if you have headache, you can just walk to a medical shop and you can buy 5 different kind of medicine to cure your headache, right? So then by putting all this effort, if we can develop another drug for headache, how much impactful that would be, right? So again, like if you take on contrary, a different example, say you put all your resources, time and effort to develop a drug for sepsis, for example, because sepsis absolutely zero treatment available today, right? Then imagine if you can develop a drug for sepsis and you can save millions of people lives, right? So that has the enormous impact. So that's why what I say is like, the yardstick, what you see, you put to measure the success for the translational science is a very different than your fundamental science, right? And but like any drug discovery or any technology development involves extensive basic research. You create the knowledge on the way you develop the technologies. So that's why one need to be very cautious about picking a right problem. And the second step is like building a stellar team, right? So this is absolutely important because, for example, if you when you take about solving any unmet need, right? And this is not like a one-person job, right? Like it takes a village to solve a unmet need. So, but what happens like as an academicians, we are trained in a particular area, and over a period of time, we develop very skill set in a particular domain expertise, and we get or build a rather comfort zone in that area, right? So then subconsciously we tend to pick the problems which fits into exactly our domain expertise and with our comfort zone, but which comes into very limited options, right? So rather, when you're picking up the problem, don't judge based on only you alone can solve it, but rather if you can just contribute to it, and if a much bigger problem you can solve, then you identify, see like how much you can contribute and what are the other skill set we need, and build a team with your complementary expertise people. So these people could be from right in your own lab, and also your collaborators, build much wider scale, like anywhere now globally you can reach out to anywhere, right? So build a team to solve this. So this is absolutely important because that depend upon the team you have, you can aim for solving much bigger problems to have much broader impact. The third factor is you should start getting the constant feedback from your stakeholders or end users. So in biomedical, for example, like this could be your patients who receive the medicines, or the clinicians, or the hospitals, and so on. And it's absolutely important to take this feedback right in the beginning, because as scientists, like, we focus completely on the science. The way we put our benchmark is a very different. We have an hypothesis, we go and validate them, as if it is going that direction, it's successful, right? But end of it, the end user might need a completely different benchmark your technology should need, because they know exactly the gap that exists compared to what it is today than what it is needed tomorrow. So understanding that in the beginning itself allow you to tune yourself to develop the technologies which meet those needs. So that's why it's a constant feedback is absolutely important if you're focusing on solving an unmet need. So these are the three main factors influence how impactful technologies you develop. So what I'll do is like, I will run through some of our own examples, what we have been developing in our laboratory, and you'll see like how these factors influence to progress our research, right? So my lab at inStem, majority of my lab focus on developing novel materials for the drug delivery, right? So then the dilemma is like, what kind of drug delivery system one need to develop to have much bigger impact. So then we focused on inflammation because, as you know, like inflammation is inherent part of majority of the diseases, like including arthritis, psoriasis, lupus, and so on. And all of them are chronic in nature, right? But if you see, there are very potent anti-inflammatory drugs and immunosuppressant drugs are available today. But despite having these very potent drugs available, the overall therapeutic outcome from these patients' treatment is rather poor, right? So that is because what happens, the nature of the disease itself is highly fluctuated. For example, like inflammation, right? It's highly fluctuated for, it's not like the steady state. It doesn't come like a once and it have a steady state and disappear, right? And it has like increased phase, and you have the steady state phase, you have remissions and occasional flare-ups and so on. And it changed from patient to patient. And unfortunately, today, we do not have any reliable non-invasive method to measure the level of inflammation a patient has. So that makes it very hard for clinician to prescribe right dose of the drugs they need to take and the right duration, right? So that's where we thought, like, can we develop a completely new concept, new material, new class of material to deliver the drug specifically to these inflammatory disorders, right? So then we started focusing on that. Like if you step back and see like almost 70 years history of the drug delivery, there are two major breakthroughs have happened, like which changed the field of the way you deliver the drugs. So the one is, in the very early stage, developing these excipients. These are either small molecules or polymer based excipients. But with selecting appropriate excipient, one can dissolve your drug and administer them. With a single dose, when you administer, it keeps the drug in a therapeutic window for a certain period of time. When drug concentration goes to subtherapeutic level, you give another dose, so on, right? So that's how, like, doctor prescribes you, like whether you have to take medication once in a day or twice in a day or so on. And this is great when you need to take medication for a few days to a week or so. But in the chronic disorders where you have to take medication for several weeks to months, several months, we tend to miss the doses, right? Then what happens if you take more frequently than prescribed, then it becomes toxic in the body. But if you miss the doses, delay the doses, it becomes ineffective. So that's why, like for chronic disorders, having taking daily pills, like it's not a very effective manner. To solve this problem, late 70s and early 80s, people have developed this sustained drug delivery systems. These are including hydrogels, microparticle, nanoparticles, so on, where you can encapsulate these drugs into these biomaterials. And even if you administer a single dose, so in a sustained manner, it keeps releasing the drug and keeps the drug concentration in therapeutic window for a long period of time, so that we can reduce the frequency of administration so that we don't miss it, right? So delivering these two types of drugs actually revolutionize the way you treat the patient, right? But unfortunately, either of them really, they don't work for the treatment of inflammatory disorders. As I mentioned, if you see, this is the level of disease I plotted here. As you know, this highly fluctuated disease, right? And in that way, like when you have the nature of disease in this manner, either of them don't work, because either you end up overdosing the patient or underdosing the patient, right? So ideally, if you want to deliver the drugs, the delivery should be identical to the level of disease. Rather, when I say, like, if when you have the increased inflammation, you should start increasing release of the drugs, and the remission period, you should stop releasing. If flare-ups comes, they should able to release the drug. But as I mentioned, we don't even have the way to measure the inflammation and predict when patient have or not. It is impossible to impart this kinetics into any materials, right? So this could be achieved only if disease itself can control and decide when to release the drug and how much to release the drug. So that's the overall concept we have been developing in our laboratory for like more than 5 years now. So what is the concept is, like, first you identify a biomarker which is produced when you have the disease. Under normal conditions, you don't produce, but when the disease comes, they should be produced. This biomarker could be any biological molecule, let's say enzyme, peptide, and cytokine, and so on. And now you develop an injectable hydrogel systems or like any biomaterial which can be, where drugs can be encapsulated. You can keep them as a depot in the body, but if there is no biomarker, they don't release the drug. Okay, but when it comes in contact with the biomarker, that should able to degrade the your depot and release the drug, and release drug goes and alleviate the disease, and that stop producing your biomarker, that stop releasing the drug. So it's a feedback loop mechanism where the disease controls when to release and how much to release. So that's the overall concept what we have developed, and I will show you how this platform technology could be used for multiple medical applications, like protecting transplanted organs from rejection episodes, or increasing the overall therapeutic outcome for inflammatory arthritis and treatment of inflammatory bowel diseases and so on. So when you talk about organ transplantation, these are internal organ transplantation. There are two types, right? One is internal, where these are life-saving organ transplants, like lung, liver, heart, kidney, so on. The second class is called vascular composite tissue allotransplantation, known as VCA, where in these transplantations, your entire tissue, like full hand, full face, full leg, can be transplanted by using microsurgeries. And each color code parts already been transplanted. That means practically every part of your body can be transplanted using these techniques. So today I'll be talking primarily about the limb transplantation method, where now microsurgeons develop so much that one can do very successful single hand transplants and double hand transplantation. In the post transplantation, in fact, these patients can regain their function. They can start doing the normal chores that you can do with your regular organs. So this is incredible, right? But the question is like, is everything is okay with these patients? So unfortunately, answer is no, because as you know, our immune system is a known defense system in our body. It's like our friend. It is designed to identify a foreign material and neutralize them. That's how we are protected from the infections, right? But in these patients, actually, your friend turns into a foe, because your transplanted organs considered as recognized as a foreign body and try to reject it, right? So that's why these patients have to take the drugs, immunosuppressant drugs, to suppress your immune system constantly throughout their life. So because of that, it leads to the complication called global immunosuppression. Their entire immune system get compromised, and that keeps these patients the verge of risk. A small infection could be very lethal for these patients. So that's why there is a huge need to develop the technologies which can way to deliver the drugs in a way you can protect the transplanted organs from the rejection, but does not cause this global immunosuppression, so that you can prevent from the potential infections as well. So, and also all these drugs are taken systemically, and when you take systemically every day throughout your life, it has severe non-specific organ toxicity and so on. So that's where we thought of developing new technologies to solve this. And as you know, when you have this immune activation that leads to inflammation, then we thought can we develop this inflammation responsive material? And people have identified that and there's a very good correlation where when level of inflammation correlates, and during this inflammatory condition, they produce this high concentration of proteolytic enzymes like MMPs and esterases and so on. So we thought of developing the technologies which are responsible, or a material which can be responsive to these enzymes to release the drug when it contacts with these enzymes. So, and our overall approach is we develop these small molecules, this called amphifiles, and which have the ability to self-assemble together. All these millions of molecules come and self-assemble to form these nanostructures. And during the self-assembly process, one can encapsulate the choice of your drug, say here in immunosuppressants. And self-assembly process should be robust, that they don't release the drug in a non-specific manner. But when they get exposed or come in contact with the enzymes that are associated with inflammation, they should be able to degrade these nanostructures and release the drug in a responsive manner. So that's the overall approach what we have been developing. And to identify the small molecule which has this ability, what we did is like we focused on the list of compounds called generally recognized safe by FDA. These are the molecules have been used in variety of food products, cosmetics, and several other products which are consumed by human, and their toxicity profiles are well studied and they are fairly safe molecule to use it. And we screened several hundreds of them and came up with like 4 molecules which fits all our criteria. And one of them is triglycerol monostearate, which is like a classical amphiphile. And we optimized all the condition, developed the technologies to induce the self-assembly of these molecules to form hydrogels. And we selected the drug called Tacrolimus. This is the prescribed drug for the hand transplantation patients. And it's a known immunosuppressant. And we optimize conditions to encapsulate this drug in appropriate doses into these injectable hydrogels. Once we have that, we went and tested their ability to release the drug in response to the enzymes. If you see, this is the amount of drug that is released. If you see, like in the absence of any enzymes, this is just buffer, right? You have like up to 5 to 10% of non-specific release, but after that it stabilizes, it doesn't release. In fact, we tested up to 90 days, like we don't see any non-specific release at all from these gels. But whereas when you expose to all the proteolytic enzymes, series of proteolytic enzymes, you can see they can degrade these gels and release the drug, right? So that shows that, okay, this material has the ability to release the drug in response to the proteolytic enzyme. But these are like a nicely stabilized enzymes. Then we wanted to see whether real inflammatory-like condition also can do the same job. Okay, for that, like we took the macrophages. These are your known immune cells. And you can take rested macrophages and you can activate by treating a lipopolysaccharide. And this activated macrophages resembles your inflammatory-like condition in your body, right? And they also produce these proteolytic enzymes. So then we wanted to see whether this inflammatory-like condition can induce the release of the drug. To test that, we took the condition media from this activated macrophages as well as non-activated macrophages, like in healthy conditions, and exposed to these gels to see the release profile. Okay, and when the gels were exposed to the non-activated macrophages condition media, the release was like almost similar to PBS, right? Like a buffer. But when the gels were exposed to the condition media that was taken from the activated macrophages, like means like inflammatory like condition, you can see the responsive manner they release the drug. Not only that, when you keep giving the fresh bolus of this activated condition media, you see the responsive manner they start releasing the drug, which is clearly suggesting that this material can respond to inflammation and release the drug in a dose responsive manner. Okay, so once we have that, then we went and tested their ability to actually protect the transplanted limbs from the rejection. Okay, and for that, like what we have done is we did entire limb transplantation kind of completely mismatched rats, from brown Norway rats to Lewis rat. And the post-transplantation, this is how it looks. And we can take these injectable hydrogels and inject the gels subcutaneously under the skin, keep them as a depot. And you follow this entire survival of the graft. Okay, I'll just quickly summarize the results. For example, once you do the transplantation and the post-transplantation, if you don't give any treatment or inject only gel and not with the drug, okay, and you can see like within 11 days, more than 50% of the animals start rejecting the graft, because you can see the sign of inflammation and so on, and grade 1 rejection starts. But whereas as a control, you take the just drug, tacrolimus, and do the local injection and without putting into gel, okay? So in that case, you can see within 33, survival is only for 33 days, because after 33 days, they start rejecting. You can see a severe muscle damage, tissue damage, and severe inflammation and grade 3 rejection and so on. Now what we do is take the same dose of drug and put it into our enzyme responsive gel and inject them. And this is the post transplantation on one single injection. And as you can see, you don't see any sign of inflammation or rejections, and the hair grows back, animal keeps healthy. And in fact, over a period of time, it starts gaining its function. Now it's able to like crawl and start moving its limbs and so on. And we also tested their entire systemic immune system is active, and there is no non-specific organ toxicity at all, and so on, which is like incredible, right? then we wanted to see how long we can push the survival of the graft, right? So for that, like as I mentioned, if you take just drug without having any gel, with a single injection, you have 33 days survival. Same dose of drug, if you put it in the gel and give one single injection of the transplantation, survival goes to 190 days. Actually, the gold standard in the field is like even including any side effects, if you can reach to 60 to 80 days is a gold standard. But like with a single injection, we surpass that. And not only that, then we have done extensive pharmacokinetics and saw how long drug is present and so on. So based on that, then we did the periodic injections, like, after transplantation surgery, do day one injection, after 3 months you give another injection. Survival was extended to 260 days without having any side effects. So which is clearly suggesting that if you do periodic injection, you can protect the graft as long as animal is alive. So that would be reminiscent to like in human also, we might able to give these periodic injections and protect the transplanted limbs. Okay. So then another major idea is like, whether it can prevent your opportunistic infections that are normal with your standard care, what they receive today, right? So for that, what we have done is like we did transplant 2 group of animals, and first group of animals received our inflammation responsive gels. Like, every 70 days, 1 injection was given, and for 4 injections are given, and study was terminated on 280 days. But whereas in the second group, all these animals, they received like a standard therapy. It says oral therapy they received, and much lower doses, and every single day for 279 days, which is similar to how patients they take medication today. And in both cases, if the rate of survival of the graft is same, but every animal that receives systemic therapy have developed opportunistic infections, because since your systemic therapy and your entire global immune system compromised, so that led to opportunistic infection. But whereas none of the animals that uh, received this localized and inflammation responsive material didn't develop at all any infections. So which shows the great advantage of the technology that has been developed here. Okay. Once we have that, we went and tested their efficacy in larger animals as well, in swine. And because swine is the robust model to just the rigorousness of the technology, and they also it worked very well. Now we're working with all the clinicians to de-risk further, to optimize further, to go into the human clinical trials soon. So with that, it brings to our second examples where we can use this inflammatory responsive localized delivery mechanism for treatment of inflammatory arthritis. As you know, arthritis is primarily localized joint inflammatory disorders, and there are several potent drugs are available today, but all of them they have to take systemic, like Vix and Bextrandol, and but need to take like large doses in order to have good efficacy at the site of inflammation, which is in the joints. But like since these are very potent drugs, they have shown like a plethora of side effects. Because of the severity of the side effects, the FDA has recalled several drugs, like including Bextra and Vix from the market because of their side effects. So then simultaneously, what people have started doing is, instead of taking systemic delivery of these drugs, people started using directly intra-articular injections, to the local injections of this drug directly into the joints, right? But unfortunately, it wasn't very effective because once you inject this free drug there, they diffuse from site of injection, right? And their half-life residence time there is a very poor. And there are a series of drugs have been injected in this manner, and an average of like half an hour to 5 hours maximum they stay there and disappear. So, and you can't do frequent injections, because according to protocol, this injection you can do only twice in a year, right? Otherwise, we end up damage the joints as well. So then what people have started doing, like instead of injecting free drugs there, they started encapsulating into biomaterial, like microparticle, hydrogels, nanoparticle, and started injecting them. But unfortunately, here also the overall output wasn't that great, because these materials, irrespective of whether there is a disease or not, they continuously release the drug for a few days to weeks and disappear, right? But as I mentioned, like, inflammation is highly fluctuated, right? So that means we might be delivering the drugs when not needed, but when disease comes back, you don't have drug depot to respond to that. So that's where we thought, like, maybe having this inflammation responsive material would have like a much better therapeutic advantage in the treatment of arthritis. So then to test this, what we have done is we took the triamcinolone estone drug, which is TA, which is prescribed drug for arthritis, and again optimize conditions to encapsulate into this inflammation responsive hydrogels. And once we developed that, we went and tested their ability to release the drug in response to the enzymes, as I mentioned. And also, they also release the drug in response to inflammation, as shown here. But more importantly, we wanted to see, because in arthritis patients, right, like your entire synovial fluid that present in the joints becomes inflamed, right? Then we wanted to see whether the inflamed synovial fluid can also release the drug in a responsive manner, right? So to test that, what we have done is we collected the synovial fluid from arthritis patients as well as healthy volunteers whose synovial fluid is not inflamed, and expose these synovial fluid to our gels to see the release profile. Okay, as you can see here, if you see this yellow line, where when the gels were exposed to the synovial fluid that was collected from healthy volunteers, it did not have much different release or extended release. The release is as good as in a PBS. The underneath the black line you see, that is the PBS, right? So that means your synovial fluid doesn't have really influence to increase the release of the drug. Okay, but whereas the synovial fluid that was collected from arthritis patients, these are inflamed synovial fluid, when you expose these gels to them, and you can see the release of the drug. And at fresh bolus, whenever you go, you see the jump in the release. Okay, so which is clearly suggesting that indeed the inflammation that present in the joints are the cause to release the drug in a responsive manner, right? So then the second important factor we need to see is if there is no disease, whether these drugs can or these gels can retain at the site of injection, and whenever the disease comes, can be able to release the drug. Okay, to test that, what we have done is you take the healthy animals and inject these gels. These are like fluorescently labeled gels, and inject them in the joint and observe over a period of time, like up to 2 weeks, and you don't see any much reduction in the signal. And these animals are like a free animals, right? They can move around. So that means despite of their mechanical stress, they don't release the drug and they remain at the site of injection. And then if you induce arthritis in these animals, that we can do by injecting KBXN serum about 37 microliters, and which is known to induce mild arthritis from day 4 onward, day 6 onwards. Okay, and as you can see, once you inject this serum, like after 4 to 6 days, you can start seeing the disappearing of the signal, which means once it start developing the arthritis, they start releasing the drug. Okay, but instead of 37 microliters, if you double the dose, like say 75 microliters, if you inject, that known to induce severe arthritis from day 2 onwards. Okay, so when we did that, you can see right after 2 days, a fast dissolution of these drug gels to release the cargo, which is clearly suggesting that this material has the ability to remain stable without releasing the components, despite your mechanical strength, if you don't have the disease. But whenever the disease comes, they can respond and release the drugs. Okay, so once we have that, we went and tested the overall therapeutic outcome from this treatment. And then what we have done is we induced arthritis, then you inject either free drug or the same amount of drug encapsulated into this gel and inject. And if you measure the overall therapeutic outcome, one way to measure the swelling of the paw, the thickness of the paw, the less swelling is, the better therapeutic outcome. As you can see, when you encapsulate a drug present in the gel, has much better therapeutic outcome compared to the same dose of drug that was given free. And the same thing corroborated by clinical score as well. So which is clearly suggesting that indeed this material has the ability to protect or increase the overall therapeutic outcome by having this inflammatory responsive biomaterial. So that brings to our third example, which is like how we can use this inflammation responsive material for the treatment of IBDs. But we added another layer of design here. It's not only inflammation responsive, but these are inflammation targeting as well. So which is very important in IBD. For example, like as you know, like IBD comes in two variants, Crohn's disease and colitis. Where in the Crohn's disease, your small intestine and large intestine both affected, but whereas in colitis, the patches of inflammation you see in the colon. So again, like as I mentioned, like either in colon or small intestine, it's not that entire colon becomes inflamed, right? You have patches of inflammation. And today there is no way to deliver the drugs to only site of inflammation. When you give the drugs, the same dose can consume by healthy tissue as well as your inflamed tissue. And unfortunately, a side effect of this, like these drugs can convert your healthy tissue into inflamed tissue as well. So that means like overall, like there is a balance, like your healthy tissue is becoming inflamed becoming healthy. So that's why the overall therapeutic outcome is not very, effective. So that's where we thought, like if we can develop a material which can be stable in entire GI tract, but which can sense and adhere to the inflamed tissue specifically without interacting with the regular tissue, your non-inflamed tissue, and release the drug only to the site of inflammation, might have much better therapeutic outcome. We can eliminate all the side effects. But the question is how one can induce this specific adherence behavior only at the site of inflammation, right? So, but if you see, when you have the inflammation as a defense mechanism, our immune system start producing high concentration of antibacterial peptides and bactericidal proteins, and all of them are, they're cationic peptides and cationic proteins. And because of that, this entire inflamed tissue becomes a cationic charged bed, right? So we thought, like, can we use a simple coulombic interactions, ionic interactions, means to induce the adherence at the site of inflammation? So you take the particles which are complementary charge, so that they don't interact with your healthy mucus, healthy tissue, but wherever you have the inflamed tissue, they can go and adhere them in a specific manner and release the drug. So that's the overall concept what we have been developing. So again, like we selected another grass-based amplifier, which can self-assemble to form these microparticles and can encapsulate the drug. Here, the drug is dexamethasone, which is known drug for the IBDs. But this amplifier also should have the charge, so that the overall particle charges should be the negative. And again, they should be able to release the drug in response to enzymes and so on. And the amplifier which was selected is ascorbyl palmitate, which is nicely amphiphilic and also renders a negative surface charge. And we fabricated optimized conditions to develop this microfibers, and you can see all these fibers with the drug, without drug, imaging agents, all of them, they have this high negative surface charge, so that they can bind to the cation surfaces. And then we went and tested their ability to release the drug in response to inflammation and so on. But more importantly, we wanted to see whether they can able to bind to the proteins and all that are overexpressed at the site of inflammation. So for that, what we have done is you take this dye-label microfibers and you take the glass plates where coat with the mucin. Mucin is overexpressed at the healthy tissue, healthy mucus, and transferrin is the one overexpresses at the site of inflammation. So these coated plates were incubated with these fibers and washed extensively and imaged them to see how much fibers they attach. And as expected, we can see all the fibers, because of their negative surface charge, they can bind to all the cationic peptide or cation protein coated surfaces, which is very logical, sir, nothing surprising. But the question is, can we get similar level of specific adherence targeting manner in vivo conditions? So to test that, what we have done is we take the mice, healthy mice, and give them dextran sodium sulfate for 7 days. and they develop the colon, IBD, which is like inflamed colon, right? Now you take these fibers and through enemas, you administer them. After 24 hours of administration, you take out the colon, surgically resect them, and image them to see how much fibers are attached on this colon. Okay, and if you see here, all the healthy animal, like which doesn't have any inflammation, if you see these colons, none of the fibers are really adhered them, because this is after 24 hours, right? You have high bowel movement, they completely might have excreted already. But on the contrary, when you have the inflamed colon, you can see large patches of this fluorescent microparticles adhere to the site of inflammation. And this is despite of your high bowel movement, they still remain there. So which is clearly suggesting that this material has the ability to specifically go and adhere to the site of inflammation to release the drug. So once we have that, we went and quickly tested their overall therapeutic outcome, treatment outcome. And one is can be measured by histopathology score. The less score is the better therapeutic outcome, right? Less inflammation. So, and again, like we tested with the same amount of drug that was given, either free drug administration or encapsulated into this targeting material and given. As you can see, you have much better therapeutic outcome when you targeted manner you give them. Right. So then the question comes, although we have shown this specific adherence into rodent model, then can we able to get similar adherence in human tissue as well, human inflamed tissue as well? So to test that, what we have done is we collected the biopsies. Through biopsies, we collected the tissue from IBD patients. From same patients, we collected tissue at the site of inflammation, as well as the healthy colon. Also, a piece of colon was taken, and ex vivo they were incubated with these fibers and extensively washed to see whether they can adhere. As you can see here, the inflamed tissue has much better adherence with these particles compared to the normal tissue. For example, even individual patients, if you see, a patient like a normal tissue, say it has an X amount of adherence, same patients in the inflamed tissue, you see much higher adherence. And the same thing, it goes true with every single patient we tested. So which is clearly suggesting that indeed, this material has the ability to go and specifically bind to the inflamed tissue in a specific manner. So that like we anticipate that like because of this particular behavior, we might be able to completely eliminate the non-specific release of the drug and all the side effects that are associated with them. So we're putting now we are taking this further for the clinical trials pretty soon. So just to summarize of this part, like, having this developing disease responsive and disease targeting biomaterial as a platform technology could able to show that they are able to protect the transplanted organs from rejection episodes, and also can improve the overall therapeutic outcome in the treatment of inflammatory arthritis and IBDs, right? But if you step back and see like our 3 parameters that can influence developing technology, do they fit it here, right? If you see like all of them, they're individually, they are like a huge unmet need. There is like a constant billions of dollars poured into developing technologies to solve these needs. So definitely there are good unmet needs. And also like the team, like, we immensely we collaborate, like because we know that like there's a lot of complementary expertise we required. So, and this expertise we build within our lab itself, like we have like a wide background of the people, with a very diverse background of people we are there. At the same time, we also collaborate with a very complementary skills people. And for the feedback, like and for each of them, like we extensively work with the clinicians, because they know like what are the gaps exist in the treatment when they treat the patients. So getting that constant feedback from them, it enabled us to reiterate our technologies that we're developing and coming refine them to which will be the clinical ready technologies. So with that, maybe I'll take like less than 5 minutes and quickly give you another example where recently we focused on developing germicidal antiviral mask to protect from the viral infections. As you know, like during this tough times, this pandemic has taught us several things, and like, it has become inevitable to, and it became the norm to wear the masks and all, right? We all wear single mask, double mask to protect ourselves. And but if you see like all masks today, what people use, all these masks act as a filtration devices, right? You take N95 mask, surgical mask, or regular 3-ply cloth masks. All of them, they work in the principle of filtration. They're efficient filters, right? But there's a lot of clinical studies very recently have shown that, with published in Lancet and all. People have shown that uh, including viruses and bacteria, including SARS-CoV-2, which is causing our current pandemic, can remain alive and active on this fabric, and up to 7 days, and different surfaces they can remain active there. So which means like it can, this mask sometimes it can become some of the reservoirs where you can accumulate of them. That's why it's extremely important to not touch and so on when you are wearing, because if you have this accumulation of this, when you touch them and again you touch your eyes and mouth and all, like you can get infection. So that's why one need to be very extremely careful. So then we wanted to see like to just establish how much bacterial or virus accumulation happens on this mask. So to test that, what we have done is, for example, this is a typical agar plant growth where when you don't have bacterial colony, this is a very clean, transparent plates what we see. But if you load the known amount of bacteria and incubate them for 24 hours, you can see this completely foggy kind of cloud kind of system here, which shows the entire bacterial colony growth, right? This entire plate is full of bacteria. So now what we have done is when people are asked to wear like, N95 mask, surgical mask, cloth masks, which were used for like 6 hours, and took those masks after they use and touch on this plate and left them for 24 hours to see the bacterial growth. As you can see, all these masks actually have the live bacterial growth, and we can see extensive amount of bacterial growth on this. So which is suggesting that indeed they are efficient filters, they can stop, but there are bacteria or virus that can remain active there. So that you should be extremely careful when you are adjusting or handling your mask. So we thought like, is there any way to deactivate them, all the bacteria or virus that has accumulated here, okay, so that we can prevent our potential infection that comes with the contact of your masks and so on. So for that, what we have done is like we wanted to develop a small molecules. These are a new class of molecules called Quaternary ammonium salts, and we have extensively optimized to have the better germicidal properties. The principle how they work is like, because all these bacteria and the viruses, especially enveloped viruses, they have a membrane around them to protect them, right? So these molecules are designed in such a way that these molecules can interact with the membrane of these enveloped viruses and bacteria and physically rupture them, okay? So due to that, you can deactivate all the bacteria and enveloped viruses. And then what we have done is we have optimized the right molecule, which has the efficient antibacterial and antiviral activity. And we optimize conditions to keep them on the fabric, covalently attach them on the fabric. And this activated fabric, we call germicidal fabric, can be stitched into variety of PPEs, right? Like mask and clothes and so on. So once we developed that, we went and quickly tested with the efficiency of them. As you can see in your regular mask, as you can see huge amount of bacterial growth, right? Then what we have done is the masks that were made with the germicidal fabric, we call G99 plus technology. And we asked people like 6 hours, and also they were used for 18 hours. And after 18 hours use, again, they tested in the same manner attached to your plate and asked them to grow 24 hours. And you can see absolutely no growth of any bacterial colonies and so on, which is clearly suggesting that indeed this material has the ability to deactivate any bacteria that comes in contact with it. Okay, so then what we have done is Then we went and showed this, and we transferred the technologies, went for next level. And so just to summarize, these are some of our efforts where we focus primarily on T0 phase, like where identify unmet need and come up with appropriate technologies and do extensive basic research to develop the technologies and validate them and make. Then we are also equally interested in focusing on the next stage, which is the entrepreneurship program, because this is a critical step to take this technology that have been developed in T0 and further refine them to take to the next stage with the clinical development stage. We are extensively involved in that as well. Just to summarize in a minute, like for all the technologies, the inflammation responsive, inflammation targeting technologies, we put together a company in Boston, it's called Alleviate Therapeutics, that is taking to the next level now. They have done entire preclinical safety studies, toxicity studies. So they completely de-risked now. All the IND enabling studies have been done. In fact, as we speak, like we're going for the clinical trials using these technologies. For the second technology, for antiviral technology, where again, like we partnered with a company called Color Threads and licensed our technology there, and they further developed and they came up, they developed the commercial products like antiviral masks and other apparel, like since now the travel and started and people are going to gyms and all, like having this antiviral suits also might reduce the level of load and so on. So these are some of our efforts in the entrepreneurship area to take the technologies that have been developed at the T0 phase and push them to the next level. Okay, so with that, I will like to stop here, and I will like to acknowledge the bunch, like great people like our the postdocs and PhDs and JRFs and interns who have been relentlessly working to develop these technologies. And also, I would like to thank all the collaborators. As I mentioned, it takes a village to solve these problems, and we extensively work with the complementary expertise groups, and majority all of them are like clinicians, so the practicing clinicians, and they bring a lot of input to developing these technologies. And I would like to thank all the funding agencies and DBT and inStem for the co-funding, and various other funding agencies which enable us to do our work. And I would like to thank all of you for your kind attentions. And maybe we can continue in the discussion part now. Thank you so much.
Thank you very much, Praveen. You took us through, I think, a very, very huge spectrum of work in a very short span of time. Before we take on questions, because I see that some have already come up and I'm sure many more will follow, I just wanted to ask you, because in fact, most of our participants are students from undergraduate, postgraduate, science stream. I think probably with an emphasis on biology. What are the profiles of the students coming to your labs? Because I'm sure there must be a curiosity to learn if one wants to engage in such or partner in such efforts. Is there any special training? Is there a specific background that works better than others?
In fact, like we are very open in terms of the background of student. Because our lab students, they come with, they are like
not just your lab, Praveen, sorry to interrupt. I would say in this area in general, what are the kinds of backgrounds that this kind of work attracts?
Oh, I see. So, I think the basic fundamental knowledge in chemistry and basic knowledge in biology is sufficient. In fact, people can transition from the biotechnology background, classical cell biology background, and the synthetic organic chemistry background. So it's all about if they are focused on the translation, and one can, if you have the just basic chemistry and basic biology knowledge is sufficient to pick up the skills. And once you start working, you need to have the very specialized domain they start reading and developing. But as long as you have either biology or chemistry or biotechnology background, one can start their career in this.
Great. Okay, we'll start taking questions, and I encourage our participants to please pop your questions into the Q&A box if you can. Identify yourself and your college, but if you don't want to, as we've always said, please use the the option of writing in a question anonymously. Praveen, the first question comes from that I'm taking here comes from Viraj Vishnu Prasad, and what is being asked here is that if the chemical mediators of inflammation are produced at a distant site, like in a tumor, will the disease-specific biomarker bind and recognize those sites as well?
Yeah, very good question. Answer is yes. In fact, like one of our goal is to develop the material which can respond to the very distant inflammation as well, which is extremely important if we want to extend this technology for the internal organs. For example, like when people do like a kidney, heart and lung transplantation, doing local injections becomes highly invasive. It's impossible to do that. So our idea is like if you can keep them as a depots even far in the subcutaneously, can able to sense the systemic inflammation and release the drug. That's the question we asked, and it does. For example, how we tested is, for example, when we did the limb transplantation, right? If you do the right limb transplanted, one way to do is inject the drugs or the gels in the same limb which has been transplanted. But instead of that, what we have done is we have injected into contralaterally. Means like if you do right limb transplantation, we injected in the left limb, and we could see that the systemic inflammation that developed, these gels were able to respond and release the drug and protect the graft. But the efficiency was like about like 70% compared to the direct local injections. That means theoretically it works, and if you refine further, we can have much better systemic responsive sites material as well.
So there's several questions coming up. As you can imagine, Praveen, this area has always evoked a lot of interest when you speak about it. Just before I go into the questions, I just wanted to ask you, so the hydrogels that you design, for instance, in the arthritic context, as opposed to, let's say, something that may be internal, does the chemistry differ? Because one is more in the surface and the other is going, into the gut. What is the kind of thinking that operates there?
Yeah, so one is that's where the choice of the base material like amphifiles plays a major role, because once you inject inside the body, like either you go through the gut or like localized injections, they degrade over a period of time. Once they respond, they degrade, and your entire quality will be decided on the metabolites or the degraded product that comes, like what is the toxicity or safety profile of this material. So that's why it's extremely important to identify the material which you de-risk them and come up with the material which have the complete safety profile even inside the body when it completely degrades it. So that's one of the major reason why we focused on known GRAS agents from FDA, because these studies, the safety profile, toxicity profiles are well documented in the clinical. So if so that like we know like a given dose of the material, what is the toxicity profile. If you take completely unknown material, you might have a better efficacy, but then we might end up spending a lot of time and effort to show the safety profile of that. So that's why the choice of material makes a lot of difference, and that's why we cautiously we take the try to develop the material which is already the safety profile is known.
Right. Okay. So then there's a question here from Aman Shivkumar to ask that if the transplanted limb is affected by a different disease in the future, will the overall effect of the immune system, because presumably it's suppressed by the drug that has been given, will the overall effect of the immune system on the transplant now also impact management of the disease in the limb? Baba?
That's right. So I think that is one of the limitation what we have today, because uh, as I said, like your rejection episode also caused inflammation, and but whenever the infection happens, that infection also induces inflammation. But right now, these biological signatures are so similar. We don't have a mean to differentiate between inflammation that has arise from in response to an infection versus in response to a transplanted organ. So in a short answer is, yeah, when you have the infection also, it can release much higher dose of the drug to suppress the immune system. So that's the caveat what we have with the current technology what we have. So I think ideally, if we can differentiate the inflammatory signatures between infection associated versus your rejection associated, then one can design the better material which can be specific to only rejection episodes. But as of now, with the current systems, we don't have that separation. So it does
Right.
impact in that case.
Right. Okay, so question now from Alan Strehar, who has really appreciated your talk, and then says that with respect to Crohn's and IBS, the steroid, the dexamethasone intervention increases blood glucose levels and also raises other issues. Would the approach of localized delivery reduce that? And have you looked at this in your animal models? Experimentally, has this been assessed?
We anticipate it should reduce, but like we haven't looked at the blood glucose level. We haven't seen in that that angle. We were primarily focusing on overall systemic organ toxicity as well as your entire tissue toxicity, where the non-specific manner when you release the drug. So which we could completely eliminate, but like we haven't looked at the blood glucose levels. Maybe I can look into that in the next phase. So
Correct.
thanks for
I
finding
suppose
out.
you have the serum that you can go back and check, right?
That's right.
All right. We have a question now from Glavin, I hope I'm saying your name correctly, Rodriguez, who's a teacher at the St. Aloysius College in Mangalore. The question here is how now this is about the masks. Okay, we'll come back to the inflammation. I can see some more questions popping up on inflammation, but this is about the masks. The question is, how effective is this material with regard to the inactivation of the SARS-CoV-2 virus, and have any clinical trials being performed? Clinical trials would not be performed for this, but there would have been tests. So, Praveen, I think the question is about SARS-CoV-2 and the mask.
Yeah, short answer is yes. Of course, these are not clinical trials were done, but what we collaborated with other institutes like an RGCB and all, we have shown that the clinical isolates of SARS-CoV-2, by using this technology, we could completely deactivate them. Within like few minutes of contact, we can completely deactivate them. So we did show that in response to SARS-CoV-2, this technology can deactivate them. Yeah.
Okay, so we're going back to the controlled delivery of the drugs, and this is the inflammation component. So there's a question here which is to ask that how can the current inflammation responsive drug release platform be extended to internal organ transplantation? Are there any insights on this, whether the depots can be maintained in such cases?
Well, as I mentioned before, like it is possible, but we need a lot more further refinement in terms of the efficacy to have this sensing far away inflammation or systemic inflammation to respond. We are taking two approaches for the internal organs. So one is, can we, during the surgery itself, can we have enough depot which can be implanted there itself, so that your entire first major rejection phase can be tackled with that, and subsequently you aid with this local injections or subcutaneous depots, which is sufficient to maintain them. In fact, we have tested with, we collaborated one of the liver transplantation surgeons, and we tested that having this subcutaneous depots we could still able to protect the livers. But as I mentioned again, the efficacy was like about only 60% compared to your systemic therapy. But 60% is huge when you, without having any systemic immunosuppression or systemic therapy. It's only local therapy for the internal organs. 60% is, we see like a really great hope we have. So we are further refining this technology to reach to push as much as possible, but it is possible in the sense.
Great. Praveen, just a question, because there's some more in that domain coming up. You went very quickly over the identification of a biomarker, which would, I'm sure, be quite a big challenge. What does it take to identify the right biomarker? Because biomarkers are not unique to just that particular space. Just your thoughts on what the challenges have been there?
Yeah, I think again, of course, it is a huge challenge process, but on the contrary, for majority of the diseases, this is fairly established in the sense, like, for example, for inflammation, the proteolytic enzymes as a biomarker is like a very well established. Like that, like for each type disease, there's a lot of knowledge already available if one can pick from them, and designing is better. But if it is not available, yeah, I think finding appropriate biomarker is the huge task, and that would be the rate determining step. And also it happens that majority of the time, for majority of the diseases, biomarkers, it's not very specific to that. For example, if you take proteolytic enzymes, right? Even under very healthy conditions, you have some basal level of proteolytic enzymes still present in the body, right? So, but when you have the inflammation, you see elevated levels of this one. So that's where the challenge comes. So then you have to design material or like optimize in such a way that the base level of proteolytic enzyme shouldn't degrade and release the drug. Okay. Only when you see this elevated level, it should cross a certain threshold, then only they start degrading and releasing that responsive. In that way, we can avoid non-specific release of them. So in other words, it's a case-specific manner we have to design. Well, like each disease, like you need to case-specific manner design and optimize and develop. But the identification of biomarker would be the far more important.
Well, far more, but perhaps really important to get it right. Yeah. Okay, so now we have a question from Anjali at St. Joseph's College, and thanking you for the talk. Praveen, I should say that almost everyone who's posted a question has really appreciated how effectively you put across, I think, a very complex field. Again, the question is about management of tumors. And the question being asked is it possible to use this method to locally enhance the immune system activity at the site of a tumor, as opposed to the use of drugs? So for the instance, the person asking the question is that could the gels be combined with immune cell activation so that it could function in therapy?
Conceptually, it is feasible. Conceptually, it is feasible, but one need to dig in a little more details into like exactly what immune activators you want to put it, like how you want to design to release and interact with the local system. That one need to design in a more careful manner, but conceptually it is possible. It is.
But how about what is being done with MSCs in limb transplants, right? Aren't they attempting, and in wound healing, isn't something like this being attempted?
Yeah. So in MSCs, in fact, like one of the challenge was
we should say that MSCs are mesenchymal stem cells, right?
Yeah,
I mean, just
that's right.
Yeah.
Yeah. In fact, like they have very good response to say wound healing or inflammation, but one of the biggest challenge with MSCs, especially like cell therapy, when you inject them, the biggest challenge was the homing capacity, because they don't have much targeting ability or homing capacity to the site of inflammation, because then you see less than 1% of cells are actually home there. So some of the approach what people have taken is to increase this uh, accumulation of injected MSCs at the site of inflammation. They have surface modified the MSCs. For example, when you have this inflamed epithelia, um, you have this E-selectin sandal overexpressed there, and people have put the corresponding ligands on the engineered cell surface, like like selectins and all. Then when you injected this surface modified MSCs, and you see large amount of injected MSCs home to the site of inflammation, because you induce the first interactions between the inflamed epithelia and these MSCs, and that leads to the transmigration and have the much better efficacy in that manner. So these are the bioengineering techniques people have been following in terms of cell targeting.
Okay, great. There's not a specific question to you, but there's a person who's asked for a feedback link. We are just going to provide it to you. As I've been saying in the earlier seminars, I'm sorry I forgot to mention it today. We have a dedicated email ID, which is sciencesetu@instem.res.in, which will just be posted for everyone to provide feedback. And we look forward to hearing from you. Question now again, Praveen. A person's asking that what are some of the problems that arise while transitioning from the preclinical phase to the clinical phase?
Okay, so, well, there are numerous. So one is like, that's where like I will go from backwards. Like as I was mentioning, getting feedback is super important, because if you want to go to the clinical stage, one need to have a very stringent benchmark people put cut off to see whether that technology that have been developed at T0 phase actually meet them. Okay, so once you meet them, the challenge to take from T0 to your clinical development phase is de-risk them, because in the T0 phase, primarily what we focus is like, whether or the concept works and have the very efficient proof of concept in animal models and so on. But one of the major criteria which is not addressed is like, having very detailed toxicity profile and safety profile of technologies that have been developed. So in that development period, like this is called IND enabling stage, where one need to understand, for example, if you're developing a new drug, one need to understand extensive manner the pharmacokinetics and pharmacodynamics of this drug, and what is the maximum tolerated dose, and like what is the minimum effective dose. All those things we need to see, and see, just to give a ballpark, right? Like if you tomorrow in patients, like a 1x dose you are planning to inject, you need to show the toxicity or like safety profile 30x of that concentration in animals, show that like it doesn't have the toxicity. So which is huge. And all these parameters, although many of the drugs are effective, many of them don't meet this criteria. They drop off of the pipeline at this stage because of all these parameters. So it's basically de-risking is the major activity that will be done during this stage, which can take to the clinical phase. And in the clinical phase is primarily to do the increase the large cohort to show the efficacy and show that it's not toxic.
Brilliant. Okay, I think we have gone almost half an hour into questions, and I think since there are also no new questions coming up, perhaps we can close the session today over here with thanking Praveen again for a very stimulating morning. And of course, to all our participants, thank you very much. And we look forward to seeing you at our next session. So with that, we can now close this meeting. Thank you.
Thank you. Thank you all.
Thank you so much. Alvin, you may close the call.
Thanks, Alwyn.